DNA structure. We're doing a good job reviewing that serial chromosome structure. Totally forgot somebody asked about the 8th genome, and I got to go back and still check that out. They were talking about coding regions or non-coding, so it's still on my to-do list. Sorry. Nothing. All good. Okay, so today we'll do a quick overview of DNA replication. But again, this should be mostly review. I won't go back and test you on what are the enzymes in DNA replication, draw a replication for. Again, that's been biomaterial in my book. So we're going to move on from there and look at other cool things. So here's your big questions. What kind of things affect the regulation of DNA replication, or the timing, or the process, or completion? Is it important? DNA replication? Does it occur all the time? No. When does DNA? Yeah. When does DNA replication occur?
During this,
In the cell cycle.
but the cell cycle is happening all the time at different parts.
That's not true. Not all cells are dividing. Do neurons divide?
Well, I'm not saying every
Neurons don't divide, therefore there is no DNA replication. Prove me wrong. Might be wrong, I don't know. No, my point is that it is only occurring if the cell is dividing, so it occurs in S phase during the cell cycle, right? Is it important to complete it properly, faithfully, with lots of mistakes? Like, yeah, mutations. How do you get the whole? Gamma rate, not replication. That's still DNA damage, though, right? It is crucially important, isn't it? Crucially important. I'm reading a book now where they're talking about mutation rates and if it can explain evolution and natural selection and all that. And our mutation rates are actually higher than scientists thought back in the 70s and 80s. They're actually calculating, I forget what the number is now, but it's a pretty high mutation rate, even compared to mice, which have an even higher mutation rate than we do. So that kind of brings into question our genome integrity, or the structure of the genome over time. What does that mean? That's a lot of big questions that nobody really has the answers to. Okay, but the DNA is very, very important, right? We looked back at it last time. It's the instructions. When you copy that, it's going to be very important to do that properly. Okay. So first few slides are review. You can kind of go back and look at it and remind yourself that replication is semi-conservative. So that means that the strands are separated, kind of like we saw with PCR yesterday. Separate the strands and then they're copied. Semi-discontinuous. Each strand is not made in the same direction with respect to each other. DNA and RNA, though, are always made in what direction? 5 to 3, and that's due to biochemistry, right? You have to add nucleotides to the 3' hydroxyl end. And the main enzyme is polymerase 3. How does this process compare to, say, PCR in terms of enzymes? Getting a little ahead of myself, but
There's more enzymes.
there's a lot more, right? How many enzymes are in PCR? What? Some thermostable polymerase, that's it. We don't need helicase because we use heat. We don't need primase because we use what? Primers, DNA primers in PCR. And we'll see in replication the RNA primer, right? But so the thermocycler takes the place of a lot of the other things, right, that we need. So just keep that in mind. Replication is a lot more complex with, I don't know, it was like 10 different enzymes or something like that involved, okay? But a lot of enzymes means, this is a theme. I'm getting way ahead of myself now. A lot of enzymes in a process means tight control, doesn't it? Think of a pathway. What happens if I take one enzyme out of that pathway? However, doesn't matter how. Pathway stops. It stops, doesn't it? We can have very tight control over a process just by targeting one enzyme in a process. Think about those 10 enzymes in replication. That gives us lots of targets of regulation, doesn't it? we could potentially regulate any of those or all of them. So I'll come back to that. This, again, these are review. This shows adding nucleotides to the 3' end. You don't need to know the mechanism or anything, so that's nice. Replication, of course, is divided into different steps, or at least I do it. I kind of break it down for students. There's the initiation, elongation, and then termination steps. Did anybody suffer through my Gen Bio class like 10 years ago? Couple? Yeah, so you probably remember this stuff. So initiate, this is kind of what I do for students, is break it up and then look at what happens in each step. So you can kind of do that. Termination. Really? So only 3 or 4 from Gen Bio? Wow. Okay. You still have scars? Nice. I'm just kidding. It's a tough class. I push my students hard. Okay, in initiation, you have a replication origin. So we typically have 1 in prokaryote. And probably if you're looking at a eukaryote with 100 million base chromosome, you probably have thousands of origins along those chromosomes. Okay, so that's a particular sequence that's going to be recognized by the first few enzymes that are involved with that. We'll come back to that as well. So give an origin, and then what do we get? What is the complex name for this thing that's formed? It's a bubble, right? I'm just kidding. It's not. It's just a bubble, right? They call it a bubble. And each bubble gives you 2 forks. Most organisms, but not all. Most organisms are bidirectional. So the forks will move out from that origination point there. So each fork then is semi-discontinuous, right? You're going to have a, what is this? Leading strand and a lagging strand, right? It's coming back, leading and a lagging. And so again, that complicates matters because you have, well, the leading strand just kind of starts and keeps going, but the lagging is made in all these parts called Okazaki fragments. Got to keep track of all those, right? So there's a lot more steps in here. Do we, trick question, I think this is actually not a trick, but on the tutorial, did they ask you if you have a fork in PCR?
They do ask on the tutorial. I don't even know.
Do we have a replication fork?
So
I should have asked this on top. That's a good question. Everything's something. Okay, turn to your partner and convince them. This is called pair share. This is how you meet Do we have a replication fork in PCR? Now, remember in PCR we use heat to separate the entire strand, entire chromosome. So there's no forks. You just have primers that come in and you start the elongation process right across. Yeah. Yeah. Cool. Good questions. See, I like these thought questions. Here's a cool electron micrograph. You can see the replication bubbles forming in here. Kind of cool. You actually can see it. And then they will come toward each other, and in termination, they'll stop. And then you have to have these repair enzymes that come in there and finish up and fill in the gaps, any gaps that are left in there. Cool. I'm just going to kind of go through this because, again, this is review. Here's a fork. This one shows a lot of the enzymes and where they're involved. So, again, you can separate the enzymes into their different categories, kind of get an idea there. Let's see. So things to remember, polymerase 3 is the major replicative enzyme, right? Polymerase 1, what is its role? It would be kind of, I would put it in termination. Yeah, it's going to remove the RNA. So there's primers in there that are made from RNA. You can't leave RNA primers in as part of your DNA structure, can you?
No.
No.
Doesn't roll.
Huh?
Said better than not.
Better than not to, yeah. And there tends to be a lot of mistakes when you first start putting down those primers, and this is one theory why we have RNA primers. There's a lot of, or at least a higher mutation rate there because there's low cooperativity at the beginning. So they say that, it's okay to make mistakes in that RNA primer because it's going to be removed, and DNA polymerase 1 has that exonuclease activity and replaced in a high fidelity fashion with DNA. The polymerase 1 is the one that comes in and removes that primer and replaces it with DNA and gets rid of all those mistakes.
Does DNA polymerase 3 have the same exonuclease? Like if it has a wrong base pair, we'll go back and change it.
Yes, and I think I have a slide. Let's see here, because it gets confusing. I don't know how far forward that is. Here's that slide here. So I did it because of this reason. So polymerase, we don't really talk about 2 because its role is mostly in mutation repair. But 3, it doesn't have a 3 to, wait, let's see. No, it does have both. It has a 5 to 3 polymerase and a 3 to 5 proofreading. So polymerase 3 can go backwards to fix a mistake, but it can't go forwards like polymerase 1. So the difference, polymerase 1 has this 5 to 3 exonuclease. Does that make sense? Does that make sense?
Yeah.
So that's why I put this slide on here, because it's really confusing. So the enzymes are a little bit different. So if you go back and look at polymerase 3, if it makes a mistake, it can back up and take it out and then continue forward again. But it can't remove the primer, because a primer would require if you look at it this way, polymerase 1 is going from 5 to 3, excising that primer.
And the DNA polymerase is at 4, 5, and 2. Are they also the same thing as 3, or is 1 like in the direction that they go?
I'm sorry, and you lost me with the numbers.
I heard of multiple DNA polymerases that do the same thing. The exonuclease activity that the DNA polymerase 5, 4, and 2. 2.
I know there's only 3 major ones in eukaryotes, isn't there? I haven't heard of any. These are E. coli. Sorry, these are E. coli. 1, 2, 3. In eukaryotes, they go by different names. And I have a slide on that as well. So maybe that's what you're talking about. It's 5, 4, and because it was studied in E. coli first, right? This process was studied very carefully in E. coli. So they saw the 1, 2, and 3 polymerases in E. coli. And they studied that. And then in humans, we have a whole different set, like delta, gamma, epsilon. So, which we don't have to memorize either, folks. So before you get worried about that. I don't know, they do that in biochem. There's a lot of memorization for stuff there.
So you want us to know which ones might have these
only in terms of recombinant DNA. So next week, when we get into recombinant DNA, so ways to change DNA or to fill in gaps or to make plasmids, keep in mind then that you could use some of these polymerases in some instances to help you, fix a mistake or fill in a gap, something like that. So does that make sense? So I kind of want you to remember what these are useful for, not really in terms of replication, though, just in kind of what they do. Does that make sense? Okay, so with replication, realize that the first few slides there all kind of review. So you can go back and maybe watch a good video on YouTube or Khan Academy or something just to kind of remember. But then, as we start applying this stuff to recombinant DNA, it might be useful to come back and go, hey, we can use mainly RNase polymerase 1. That's the one that's used a lot in recombinant DNA. So I'll give you some examples next week and how you can use those enzymes when manipulating DNA. Is that okay? Yeah, okay. So here, this is just showing the proofreading thing, which is it's a good thing, right? So this is DNA polymerase 3 fixing mistakes. This shows DNA polymerase 1 removing a primer. But why is it good? I mean, take a step back. Who cares? Why do we need a proofreading thing? Why is that a good thing?
Case when you're adding the template, when you're adding the template, then you can go back and make mistakes and kind of recorrect.
Yeah, just the simple fact of correcting it. You look at the error rates, at least for most of these enzymes, it's only 1 in every 10 to the 5th nucleotides or something like that, or at least those are the original numbers. So they're really high fidelity enzymes. Giving them proofreading ability makes them able to correct lots of different mistakes. Yeah. So just think about it in the big picture as well. Okay. So again, don't memorize every detail, but keep in mind some of the details here with polymerase 1 especially, because we can use it to our advantage. Okay, is that cool? Okay, cool. So there's a summary of that. Let's see what else. Oh, so here are the eukaryote or human polymerases, and you can see the different names there: alpha, epsilon, delta, whatever. And the different parts of it, these are the different parts of the what they call a holoenzyme. So it's not just one little protein, it's a huge number of proteins working together. So here, certainly do not memorize this, please. I never have memorized it, okay? But it might be useful when you read a paper. I think of the paper that we read on for Friday, they talk about core proteins versus like POL3 or POL3 holoenzyme. If you run into a term like that, you may have to figure out exactly what subunits you're including in that complex, because that could then change the function of the complex, couldn't it? By switching out the subunits or taking out subunits, that you might lose part of the function of the entire complex together. Does that make sense? Or did I just? So that's kind of why I put that there, is that you can see those, but don't memorize them. And then here are the different polymerases. So I don't know if this is what you were talking about, like polymerase 542. I'd have to go back and check what those numbers mean. But normally what you see is alpha, beta, those kind of things. And the major one here, the equivalent to poly 3 is the, oh darn, it's the delta, isn't it? Poly delta in eukaryotes. So that's the major one. You can kind of compare them to prokaryotic. So I'm trying to give you some tables here where you can relate it back and forth from prokaryotic to eukaryotic. A lot of the papers we read depends on the organism. So this paper on Friday is with yeast, and they're eukaryotes. Okay, so they'll probably go by slightly different names. Okay, now you're all quiet. Okay, let's see. Let's get into some other things. So other cool enzymes that we can use for manipulating. So just keep these in mind, I guess. So for recombinant DNA enzymes, and I'll give you a whole list of them later on, but poly 1 is useful, topoisomerases are useful, and this is a mouthful, but basically what topoisomerases are enzymes that can change the 3D shape of the DNA. So typically where you use these guys are like with gyrase. So gyrase is an example of a topoisomerase, and it's the one, does anybody remember what it does? It's out in front of the fork. Yeah, it gets rid of supercoils. And the way they do that is if you have a DNA molecule, the topoisomerase will make what's called a nick. And so it's not a person, but it's a single strand cut in the phosphodiester backbone. And what that allows the topoisomerase to do, it can actually not really unwind, but it unwinds not the helix, but a supercoil in the DNA. Or what's cool is some of these topoisomerases actually introduce supercoils. So if you just put in a topoisomerase, this is gyrase, with some DNA, what you can see is, I think it's adding, it's like over time, you can see the supercoils start forming in the DNA. Does anybody know what supercoils do to DNA structure? Like if you're running a DNA in a gel, where is the DNA more supercoiled? At the top or at the bottom? Why is that true?
It's smaller.
Right, so supercoil tends to make it a little bit smaller, right? What was talking? Darn it, now this is embarrassing. Sorry. It changes the shape one way. I thought the more supercoiled ones tended to lag behind in the gel, but maybe I'm just doubting myself. I want to go back and check. Okay. So, anyways, this picture is showing the you can actually see the differences in the mass, the parent mass of these things. And what gyrase is doing here is it actually introducing supercoils to the DNA. So that's the way it gets rid of the other supercoils that are formed during replication. So it's kind of interesting. So I'll have to go back and check it. Sorry.
According to bioinformatics.no,
Okay.
it runs faster than.
If it's supercoiled?
Yeah.
Okay. So we were right the first time. I shouldn't have doubted myself. Okay, good. Kind of cool. I like showing pictures like this, so those will frequently happen in lectures. Okay, because pictures are cool. Okay, so any questions on the expectation then for knowing replication? You guys okay with just a quick review, knowing that I'm not going to just say list all the enzymes from replication? But then we want to, you should be able to maybe compare and contrast replication and PCR since they're related. And then I gave you some ideas that some of these enzymes can be used for other things.
So as far as like 5' 3' 5' that's a nuclear that could be like, you don't want to know the specifics yet, or
just go back and review poly 1. I think that's going to be the one that we're going to see in our list of modifying enzymes that are typically used when you're manipulating DNA. And I'm going to give you more examples next. That's probably a good place to start. Okay, so I already kind of talked about this. Is replication highly regulated? Okay. That's pretty much all what this class is about. Like everything is more complex in eukaryote, kind of a mantra of mine. And regulation is part of that. There's really not much in the cell that is going on just spontaneously. Pretty much every reaction is controlled by an enzyme. Every pathway are controlled by enzymes. Even those pathways are controlled by signals. Okay. So here, going back to what we started with, when does replication occur again? Is it told to start at a particular point in the cell cycle? Yes. And at the end of the semester, we'll look at the signals that tell the cell to start cell cycle, and then the kinases that tell replication to start during S phase, right? And this is highly regulated, only occurs at a certain point when it's told to, and it can be stopped. Stopped as well if there's any issues, okay? So occurs at specific times, specific signals, we'll see that later on. Must be completed quickly. How long does it take? How long does it take an E. coli to divide under perfect conditions? 20 minutes. Replication is only part of that. It has replicated 3 million base pair chromosomes faithfully. What about humans? Human cell cycle, it's like an hour and a half or something like that. And again, replication is only part of that. Now we have to do 3 billion bases and 46 chromosomes. Yeah. So I guess that's pretty quick. Pretty quick. I think polymerases go at about like 100 nucleotides a second or something like that. If I remember correctly. And faithfully. So is that one thing that could stop or hurt the completion of replication? A lot of errors. Yeah, there is. If you remember anything about checkpoints, which we'll talk about again at the end of the semester, one of the checkpoints assesses DNA quality, doesn't it? You remember that? It's way back. So yeah, replication can be stopped and you can try to fix it, okay? I think I'm giving you kind of the answers for the next question here. Let's see. No, I guess I'm not there yet. Okay, so requires multiple enzymes. I told you that already. That's something you're going to hear again in this class. When I ask, what do multiple enzymes mean in a process? You want to think multiple targets for regulation. It doesn't mean every one of them is regulated, but it's possible. And then this talks about how many times do we replicate our DNA during replication? Doesn't it sound good to replicate more? What about what if we go 1.5 rounds? Isn't that a good thing? Oh. Cancer cells like replicating 1.5 times or less. They don't care about their genomes. But this has to be carefully controlled. You have to make sure you don't over-replicate or under-replicate your DNA. You just want it 100% complete, not 99 where you lose some, okay? So all these things are going to come into play. I'll give you some examples then. Okay, so let's talk about regulation. The primosome, remember if you hear ohm, what does that mean? Yeah, big, complex. So ribosome is a big complex of proteins and RNAs, right? The genome, all your DNA, right? So something big. The primosome here is a big collection of proteins that start in initiation. Whoops, I erased it. But they start in initiation. They have to recognize the origin, right? That's important. So remember, the origin is a sequence that is recognized by these, what are they, DNA proteins. And the DNA proteins actually will bend the DNA. And guess what? If you look at some of the typical E. coli origin, if you look at some of the conserved sequences in here, we find a lot of AT-rich region. What does that mean to you? Break apart. So less hydrogen bonding. When you bend the DNA like this, you're not weakening, but you're unstabilizing the helix, just like when you bend the bonds. And then on top of that, you have a bunch of AT regions, so that means less hydrogen bonding. So what does that help us make? A bubble. Yeah, bubbles, right? Isn't that the little fish? Yes. Not Dory, but there's a different fish. Yeah. Oh, yeah. Bubbles is the fish. She likes bubbles. Okay, so we get a bubble. Is that kind of cool? Hey, it's almost like it's set up to help us initiate replication on purpose. Yes, sequences are important for binding, and then important to help the process here. Okay, and then you see helicase comes in here, the helicase loader, and you start the process. So that's your bubble there. Okay. So think about those things that are involved in starting a process like this. You have to have the correct proteins there. They're going to be there because they are signaled to be active. So you have to be active in the right conformation at the right place, the right origin. Okay. So there's a lot of different things that go on. Just that something simple as starting DNA replication. You have to have all the characters there to start. Okay. Finally, there's the question here. Let's see. How do initiation proteins recognize the origin? No, I mean, yeah, I am terrible at this. I only get ahead of myself and then give it to you. Because nobody ever complains about that. I'm giving them the answer on my student feedback forms. I see that. He gave us the answers all the time. Too hot. Yep, something specific. Is everything working today? Good old technology. That's right, scanning for sequences, it's a good point. And that kind of goes back to that picture I showed last time. These proteins have specific interactions with those nucleotides. Remember that idea of chemical environment, hydrogen bonding, all those things kind of go into that. Okay. Any questions on that? You guys good? So if they bind in a sequence-specific manner, what do you think happens to the stability of a plasmid when you start mutating the replication of origin? Good thing? Bad thing? Most people are shaking their head no. Now, if it's sequence specific and we change the sequence, how does that affect the binding of the initiation proteins? May not bind, okay? That's exactly what this picture is trying to show. This shows a yeast. This is a yeast initiation origin, and actually you'll recognize those same symbols on the paper for Friday. So they show the yeast origin. What happens if we mutate these regions? You see the level of stability here drops drastically. And in some cases, there's no changes that you can make there where you see the plasmid to be passed down to the next generation. That's what they're measuring by stability. Okay, so that's kind of a dumb question. When you have conserved sequences or binding sequences and you change those, you mutate those, that's generally not a good thing for the function of that. What about timing? This is an interesting one. So I mentioned that origins, replication origins, fire once and only once. But do they fire all at the same time? So what's kind of cool with this, what they call a timing assay, you can actually synchronize cells. So there are ways to synchronize cells so they're all going through the cell cycle at the same time. Isn't that kind of cool? So they typically use this idea of synchronizing or synchronized cells. So you start them all in whatever, G1, okay? And then you kind of release them, right? So then they all go through G1, S, G2, and they start the cycle, okay? Now, if all the origins fired at once, and we just kind of monitored how much DNA is going on, what happens to DNA as you go through replication? So, all cells go through G1, S, G2, M at the same time. So before where do we divide the cell? What bit? Where do we divide the cell? M-phase, I would argue, divides the nucleus. Cytokinesis. Okay. So before, did you say it? Okay. Now we're talking about it. Before cytokinesis, what happens to the amount of DNA in the cell? Right, we go from one, what would we say? It goes from like, what do we start with? Diploid goes to tetraploid or something like that? How would you say that? Let's just say, can I say 1x going to 2x? The amount of DNA, is that okay for now? We're just doubling the DNA? I'm trying to say. Doubling the amount. So, and they show it here, 1N just means 1 genome to 2 genome. If everything was equal, we would see something like this. All the cells would just slowly double their DNA over time, wouldn't they? If we just took a picture, a snapshot, measured the DNA somehow in those cells, we'd always see one peak, right? All the cells are going on. What do you think? Do you think that this is what we see? All the origins fire the same way and every cell at the same exact time? Yeah, I'm really setting this up, aren't I? Yeah, no, they don't. They don't here. So what you want to look at in this picture, it's kind of goofy, but look at just the S phase cells. So these are synchronized cells. They started S phase. What is S phase again? Okay. What is my pop? And then they count the number of cells versus fluorescence, DNA, the amount of DNA. Tell me about the S cell population here. So we just took a snapshot of synchronized cells going through S phase. Are they the same? Do we see one peak?
No.
Do we see this? One peak. Don't worry about the G1, G2 peaks. Look at the S fraction. What do you see? Yeah, hundreds of peaks. What the heck's going on here? It's so interesting. What does it mean to us? So they're not firing at the same time. So what does that mean? The origins. They're all firing at what? Different times. What does that mean? We have to keep track of when they fire and who's fired and when they fired. And do we? We have to keep track of them all?
I was just gonna say that each cell has to be regulated as far as timing of replication.
Yeah, I mean, each cell has to keep track of which origins it fired, so they don't refire an origin before all the origins are fired. Does that make sense? So, have I used that term to fire? To fire means to start the bubble. I don't know if I used that term earlier, sorry. So, if you think about all those thousands of origins, you can have one origin fire and then the other one start to fire. This one can't go again until everyone's complete. Can you visualize that in your brain? If it does, you've started over replicating your DNA. Not a good idea, right? Not a good idea. So every cell, in essence, has to keep track of those origins. Which seems like a mess, doesn't it? If you've got thousands and thousands and thousands of origins. So this is one of the things I want you to stare at this for a little while this week and think about the implications and how do we control it. This just talks about how basically what they did here is they were able to separate the cells based on DNA content, and you use a process like FACS, which is fluorescence activated cell sorting. So you can actually use a computer and a laser to count the cells. It's kind of cool if you ever get to do something like this. So they go through a capillary and the computer will count them based on how much DNA is in it. That's how they did it. But if you look at the process, and we'll look at this more when we talk about cell cycle regulation. Go away. But who do you think is responsible for regulating the firing of an origin? It's not a person. It's called orc, which makes me think of orcs, like, you know. But it's origin recognition complex. So ORC works with all these other proteins. Oh, wait. Look, Dr. Carpel, there's multiple enzymes, multiple proteins here. What does that mean? Multiple proteins means multiple targets to regulate. I knew you were going to get this. Sorry. So when we look at protein pathways, it's right. Okay. So all these proteins are going to be involved, and we'll talk about it like CDC is named a cell division cycle protein because it's turned on or activated in S phase. Why? To start replication. Okay. So these are the things I want you to think about. All these complex points of regulation that have to occur in just when I said, oh, here's replication, right? There's a whole bunch of stuff that has to happen even before initiation start to have that correctly done. And then if what we're going to see is that some of these proteins are actually degraded after you start the bubble. So here's the bubble. Some of these proteins are degraded so that you can't go back to the top and start it again. So ORC actually stays there, but then these other proteins have to be remanufactured before you can start them, before you can fire those origins again. Okay. So very, very complex, folks. Very, very complex process. And I want you to think about that. How do we carefully regulate this to make sure these things are going properly? Okay. Isn't that cool? Kind of that timing assay. What do you think about that? Kind of interesting. I kind of like it. Might show up on an exam, something like that, you know? What does this mean? What does this mean for DNA replication regulation? That kind of questions, okay? Questions? You don't look impressed, sorry. I tried. Yes, sir.
Is there anything that doesn't have ORCs? Like, what came first, multiple chromosomes or ORCs?
I don't know, man. I don't know. Remember, this is just eukaryotes, right? So this is in yeast. So in prokaryotes, it's not the same process. But you're asking what came first, the origin or
Like,
the,
is there another mechanism that you can like recognize it without having a big protein complex, or did it get ORC?
oh. You kind of have to have both, don't you? You have to have something that recognizes an origin on the chromosomes, don't you? In order to start the process. That's what I would argue. Those are like theoretical questions. You can come in the office and we can just go down every like rabbit trail if you want. There's a lot, unfortunately, there's a lot of things I can't answer. So, is that really a rubber chicken? There's a rubber chicken over here as well.
That's something I can't answer.
Yeah, see, there's some questions in life that we can't answer.
But I guess we know the chicken came first.
How bored we get in class. So that's a good question, and I struggle with those. A lot of times we look at a process and it's still kind of a black box, Still kind of a black box. We don't you get put it or
If I'm getting back like this.
yeah, complex. In yeast, fork stage associated with the origin, sorry. But that can vary by say organism. So I think that's kind of what your question is saying. Are there other examples? So we'd have to go look at the different organisms probably. Okay. And then in humans, sometimes we don't even understand everything, right? Because that's why we try to use these model organisms to try to understand it. I know it's not a satisfying answer, but okay. Let's see. I want to leave a little bit of time to give kind of an intro to the paper. So let's see how many things we got. So what kind of issues that again can affect replication is kind of what the big question is for today. So what have we talked about? We've talked about just initiating the process, right? What was another one we talked about? You guys keep doing this. But what about damage? Can that affect the process? Certainly, you want to think about that. If we've got chromosomal structural issues, that can affect the elongation on that, can slow us down. There are other checkpoints, so like P53 or the BRCA genes, the breast cancer genes. So those are ones that are assessing DNA quality and making sure the genome is correctly done, right? So those are things you want to think about. I gave you some examples here of how replication forks can get around DNA damage. And some of those involve just like, okay, we have to get by this. We're just going to fill in the gap and hope we don't cause any mutations, So it's kind of the end of the, what do you call that? End of the line, kind of we call it an SOS response, right? We're just trying to get through it as well as we can. So those are things that I want you to think about with that question. So what are some things? So we have to have signals. We have to have the correct proteins there. We have to have any other issues taken care of, okay? So let's save termination for Monday. And I just want to go into the paper for just a little bit. Sometimes I think it's nice to have a little preview of that. Any questions on any of the replication stuff we talked about? No? Okay, so, and I think I put it by the assignment as well. So if you go to the paper, the replication paper, I put in the pictures that we're going to talk about, okay? So my expectation on Friday is that you guys will have looked over the paper a little bit. You have written your summary, so this is going to be a difficult one. Remember those summaries, you're trying to extract as much information out of it as you can, and then write up something that your cousin, who's like a web designer, can understand, right? Not a molecular biologist. This is going to be kind of difficult. How do you relate some of these things to real world ideas that people can relate to, which be more of a general, okay? And then when we come in, I'll give you maybe 15 minutes or so. I want you to meet in your lab groups. If you remember your lab groups from yesterday, see if you can find your lab groups, your lab mates. We can move around, meet together. And then what I'm going to do is I'm going to ask groups to come up, and everyone's going to have to participate at some point. But we're going to come up and we're going to try to go through the figures to explain what's going on in the paper, okay? So this should, I know this is a big class, it's going to be a little scary, but you get to get up in a group, so you know you can bond with your group and they'll help you get up in front of everybody. If that's a serious issue, let me know. Some people do have serious stage fright, okay? So that's happened before. But the idea here is we want to try to break down the paper and understand what they're doing. So the paper is about replication. They start to look at DNA structure. So I mentioned last time, DNA is organized in chromatin, right? Yeah.
How professional voice do you want this to be written? If it's for like, trying to explain molecular biology to someone who doesn't know anything, can I use like slang and jokes in there? Like,
Fine. I always used to think of New York Times, but if you read like a scientific neutral, general voice, you know, it doesn't have to be super stuck up or anything like that. Don't we have a couple of minutes? Yeah, I guess packing up.
It's great to take any more notes.
Don't pack your backpack while I'm talking. It's not nice. Okay, so it's the other thing to realize is these histones can be remodeled. They can be moved around. So if you have to get access to something, say like a replication origin or a promoter or something like that, you can move these things around. So this paper, a lot of the figures they're looking at, they're actually looking at what the DNA looks like and what the positioning of these nucleosomes are along the DNA, okay? So that's I think half the battle is figuring out what some of these figures are talking about, okay? So you want to think about that. You also want to think about, I put some information on DNA digestion. This is one way that you can kind of see how the DNA is structured. So DNase is just a protein that cuts DNA randomly. Where do you think it's going to be able to cut easier on this kind of a structure? Between them? Is that what you said? Yeah. So think about this. If DNA is wrapped around proteins, that actually hinders access to that, but for any protein, including DNase. So DNase tends to cut in these open areas here, not in the nucleosomes themselves, okay? So that's another experiment that they use to kind of look at the DNA structure. So I put that picture there, and then there's some data here, kind of shows you how, you know, depending on how long you treat the DNA with the DNase, you can see different units. What's kind of interesting now is when you start looking at the differences between here, they tend to be about 160, 170 base pairs, which just so happens to be the amount of DNA that's wrapped around one nucleosome. So you may want to take a look at these and see if it kind of helps you understand the figures and how they set this up. So like I just said, if you look at some of these patterns, it kind of shows that the DNA is wrapped around nucleosome. And then what we're going to do is see, in addition to that, we want to look at other proteins like ORC. We just talked about ORC and ABF. Those are proteins that bind to these particular spots, these initiation or the origins. We want to see if we can figure out where they are with respect to that. Okay. So that's very short and quick, but when we come in on Friday, you guys can work on it a little bit and we'll see. Of course, I'll help you, right? We can work through it together and try to understand. Okay. Quick questions, anybody? Okay, have a good day.